Hi guys, I'm Ashish and you're watching me on Medical Acme India. And today we are going to discuss about the biostatistics, basically data, study and sample size. So guys, what happens, we are running through the AIIMS PhD entrance lecture series, and that is basically the skill and aptitude part, that is section A. What happens section A, as you all know, is very important and it is the deciding thing basically. What happens in section B, or the subject based questions, all the students are like up to mark. So what happens, all the people, suppose they are doing like out of marking, sometimes like 40 40 40 39 39 39 out of 40, they all are getting the what does this section has the importance that it is basically deciding. Suppose what happens, all the students who are good in section B or subject basis, they are not good in skill and aptitude. So this becomes very important to tackle all the questions, that is 30 questions of section A of the AIIMS PhD entrance, and it is basically deciding. Why? Because all the students have a particular kind of thinking, particular kind of like aptitude and skill, so they are like different in handling the like aptitude and skill based questions. So this is the fifth lecture of the like an AIIMS PhD entrance lecture series, and basically we are starting through biostatistics. There are 4-5 lectures of the biostatistics. We have like covered English and reasoning. If you have not studied that part, please go and read, because I know reasoning wala video have the like kind of very least viewers. So please guys, don't take that like kind of lightly. This is very important to study about reasoning, because reasoning and English, it consists of like 5 to 6 questions, so that is very important. So guys, let's start the topic. So guys, start with the question, that is, who is the father of the biostatistics? This is not like kind of questions which is asked in the examination, but it is very important to know who is the father of the biostatistics. So I'll give the answer in the last slide. So let's move toward the data. So what is data basically, guys? So data is the information. Suppose what happens, you are seeing and observing all the things in and around you. Suppose if you are like running, you are running in like kind of gym. So what data you can get there? You can get that the number of people who are coming, how much they are running on the like the cardiovascular machine, okay, cardio fitness machine. So how many kilometers they run, what is the pulse rate, what is the BP, and what is the respiration rate, all the things you can assess, and that will be the data. So what if you are running through the like kind of suppose in the roadside footpath, then what happens, what data you can get from there? You can get from there that suppose if you come and count the number of like vehicles, basically number of cars, so you can there collect the data that how many cars are of Suzuki, how many cars of Toyota. So this is all the data. So data has not a peculiar thing, it has a characteristic that it has a parameter basically. So data is something, some information in a parametric form. Parametric say, I mean ki they have the parameter. Suppose if I am getting data of your weight, that will be in kg. If I am getting data of your height, that will be in meter. So something which is quantified is a data. Okay, so what is data? Data is set of facts expressed in quantitative form. Okay, some qualitative forms are also data. Okay, but basically all the data are in quantitative form. So why that is data important? Data is important because the biostatistics basically has an advantage of assessing that data. What does that data do? That data will give the significant value, so that we can say confirmly that this is the research which we can like extract from the data or the information. So basically the consequence of the information or analysis of the information can be done through the data. So sampling, guys, what is sampling? Making conclusion on the basis of studies of the sample. What is sample and what is population? Suppose there is a population in Delhi. Okay, so I'll say that Silampur, suppose, and South X, suppose Rohini, these all are samples. And what is the population? Population is Delhi. So sample is a part of the population. Okay, so there are many samples in a population, so that the word the mu and the x is like denoted. Mu represent the population mean, and x1 bar, x2 bar, these represent the sample mean. Okay, so sampling is making conclusion on the basis of different samples. Okay, suppose if you have got the data from Hauz Khas, Rohini, Silampur, then what you have done? You have done the average of all the things, or you have found out the central tendency of all these things. Then it is called the sampling. Sampling is doing some like procedure with the data. okay, or sample. So guys, what is accuracy? These two terms, accuracy and precision, are many times asked in the AIIMS PhD entrance. So guys, please remember it. Accuracy is the closeness of a measured value from its true value, basically. Okay, so guys, what happens, suppose if I am saying that there is like weight, average of the weight of some population is like 110, and you are getting in the like repeated measures that they are 102, 103, 105, 109. So 109, which is very closer to the true value, that is 110 will be the closer value to the true value, and it is called the accuracy. But 102, 103, okay, there are four values: 102, 103, 101, 109, and your value mean is what? 110. So 101, 102, 103 are known as precise. Why? Because they are closeness of the repeated value, and the last value is 109. So 109 will be accuracy, 11, 12, 13 will be the precision. So guys, parametric test and non-parametric test. So guys, what happens if you are having a data, then what happens, you are going to do something with the data. So that something is the analysis part. So guys, analysis is basically classified as parametric test or the non-parametric test. Parametric tests are those data which follow the normal distribution curve. Okay, suppose if you have some data and you are extrapolating that on the curve and you get the bell-shaped curve that is symmetric, then it is the parametric test. And under these parametric tests are t-test, f-test and ANOVA. This is very important. I am again emphasizing that please remember these things, parametric and non-parametric, at least example. Okay, no one is going to ask you what is parametric and what is non-parametric, but these t-test, f-test, ANOVA are basically the three important tests under the parametric test, and non-parametric tests are basically the chi-square test. So guys, let's move toward the types of data. So guys, what happens, these all things I have covered in the biostatistics class for the ICMR JRF lecture. Okay, so guys, here I am going to practicize you. There what happened, there was like 1 hour of lecture, but it will be like of 2 to 2.5 hour of lecture. In some, like 5 lectures are divided into 2.25 hours of like 30 minutes of each. So here we have the sufficient time, so we will practicize some questions from the like central tendency, from the central deviation, and all the things we are going to practicize. This is basically theoretical class where I am going to discuss about the sample size, data, study, all these things. So this first lecture is the like kind of mind makeup, and the second, third and fourth we are doing like we will do like lot of practices. Okay, so guys, we will solve numericals also. So let's move toward the lecture. So guys, data is divided into two data, categorical and continuous. Guys, this is self-explanatory. Categorical means qualitative data or featured data that can be that have the quality, that do not have like values. Continuous has the quantitative or numerical value. Continuous data has the numerical value, or quantitatively we can assess the data, that is the continuous data. And categorical has the features, characteristics, properties. Okay, so that data is the categorical data. So guys, these all things are basically, I know, but what happens, I am going to revise it all. Okay, and so this is the exclusive video on like Medico Acme India. So guys, there are three types of categorical data: nominal, binomial and ordinal. What is nominal, what is binomial, what is ordinal? Okay, so there are two types of continuous data: discrete and continuous. Okay, true continuous basically. So guys, nominal data is named category. Okay, binomial data, that is success or failure, yes or no. Binomial you have learned in digital wala system mein, on or off, yes or no. If I want some analysis and I want yes or no may answer, then it will be binomial data. Ordinal data has a kind of levels of, or you can say implied orders. Orders, I mean, if I am telling that you are having a disease, then what is the like kind of severity of disease? You will say mild, moderate or severe or extremely severe. So that is the order. So ordinal data has the order. Nominal data are basically named categories. Okay, suppose if I am going to give the name for the basically data, then it will be named data. Name will be anything, random names. Okay, you can think of names of like kind of things. Suppose if I am saying that this is a library, the first library, second library and third library, so this is a name. I can give the name and you can write the data or the value according to the name. Suppose that I am saying that like JNU has the library, AIIMS has the library, and different libraries have the kind of persons coming. So guys, what happens here, the names itself is kind of self-explanatory, that nominal data are the named categories basically. So guys, discrete data basically has the particular number, and continuous data have the any numerical value. Particular number, say I mean that key, suppose if I'm going to assess, like if I'm going to take a survey from the like railway station, then I'll say that how many like are how many passengers are basically on the platform number 1. So platform number 1 to 16 will be same. So particular numbers are allotted in the discrete data, and continuous means any numerical value is the continuous data. So guys, this continuous data, basically true continuous data, is very important. This is commonly used, okay, and this ordinal in the categorical are commonly used. Continuous and ordinal, basically these two things are asked many times, so please remember these things, okay guys. So quartiles and deciles. So guys, quartiles and deciles are basically, and percentiles also, are basically the tool which actually divides the data into 4, 10 and 100 parts. So very important here is that quartiles and deciles divides data into n minus 1 part. If quartile has like 4, so 4 minus 1 part, that is 3 quartiles are there which will divide the data into 4 parts. So similarly, deciles, that is 10 minus 1, 9 deciles will be there which will like divide the data into 10 parts. So 9 and 3, please remember, quartiles are 3 in number, deciles are 9 in number. So guys, quartile range and the decile range will be like Q3 minus Q1, okay. So guys, what happens, the maximum quartile and the minimum quartile, okay. Q3 is the highest quartile and Q1 is the lowest quartile. So the difference, it is the quartile range, and quartile deviation will be quartile range by 2, and quartile ratio will be the difference in the highest quartile minus and the lowest quartile, and the sum of the highest quartile plus the lower quartile. So guys, please remember these 3 formula. These are very important. Interquartile range is depicted in the diagram here, 25%, 25%, 25%. So 3 quartiles have divided data into 25, 25, 25, 25. Interquartile range you can see, and this is very clearly shown in the like bar diagram, okay, where you can see different colors, okay. So guys, decile range, in the similar way you can think that D9 minus D1 will be the quartile range and D5 will be the like median, okay guys. So these are very important, and these are basically many times asked in the examination. Please remember these things, okay. So guys, let's plot the data in like very random manner. So guys, what happens here, you can see that 25% of data is in the like left half, and 25% in the right half, and then again 25% in the right half and 25% in the left half. So guys, this shows that 50% and 50%, that is bilaterally symmetrical contribution of all the data set are here. So this is not important, but the important thing is what? The important thing is 58th percentile or 50th quartile or 50th decile. What is that? That will be basically median. So this, please remember this thing, that will be the median, which is at the 50th percentile or decile or quartile, okay guys. So you can see the smallest value are and the largest value are very low in number, okay. And again, this is actually very self-explanatory kind of curve, so I'm not going to go into detail of this. Please use your mind and just be clear about the diagram. This is just the diagram, that is schematic. So odds and risk, this is very important. Suppose this is an apple and you have cut into 4 parts, okay. So guys, what happens, A, B, C and D, these will be all equal part if you have cutted like kind of any biasness. So that will be 1 by 4, 1 by 4, 1 by 4. If you are going to divide into your brothers and sisters, then you want, if you are a mother or father, then you want that all the mother and father have, all the like your children have the same quantity of apple that should be given, so that they do not fight with each other. So in this case, 1 by 4, 1 by 4, 1 by 4 is basically given. So this is suppose data, and you have like divided the data into 4 parts, 1 by 4, 1 by 4, 1 by 4, 1 by 4. So what will be odd, what will be risk? So odd is basically prevalence, okay. Sorry, odd, risk is basically prevalence, where you are going to divide A by A plus B plus C, means the one data divided by the whole data, one part of data divided by the whole data. One part of data divided by all the other data will be odd, okay. Remember, in this way, data that is your choice divided by other data will be odd. OO. Other data, odd. That has the O in common. So 1 by 3 will be odd and 1 by 4 will be prevalence. Okay, prevalence is basically the number of data in the total data. Okay, so please remember in this way, this is very important. So guys, charts and graphs, there were many comments regarding charts and graphs that please clearly show all the like graphs and charts. So I am here. So guys, AIIMS basically asks that which graph is basically important and which graph is basically most commonly used. So guys, I'm going to discuss all the graphs. So this is bar chart. Everyone is familiar with the bar chart. Okay, bar chart is nothing but you are going to show your quantity of the like kind of data in the bar form, and it is basically, the important thing is here, it is 2D, not it is 3D, it is 2D. That is, the height of the bar itself will be explaining the intensity or the number of the, you can say, data points. Okay, so stacked bar chart, if you are going to extrapolate the two data in the same bar, then what happens here, you can depict two data in the same bar in the stacked form. Okay, so you can use stacked bar for more than two data in a single set. So histogram is basically the kind of bar chart that is basically overlapping with each other. So histogram is most commonly used and it is basically to show the uniformity in the data. Okay, so this is many times asked that histogram is basically the uniformity of the data, which is like the advantage of the histogram. So pie chart is basically giving you the proportion. Okay, and donut chart, these both are basically to show the proportion of the population, or you can say proportion of the like kind of things which can be shown with the depicted with the pie chart. Okay, so line graph is basically the correlation or regression kind of thing, and the line graph is basically most commonly used. You all are aware that it has the x-axis and y-axis, and this is basically the relation between the x and y-axis data. Okay, parameter basically. So guys, this is stacked line, this is the same thing, line graph and stacked line graph. Again, you are here using two parameters, or you can say two data set, and you are extrapolating one on another. So pyramid chart are basically used in the, you can say, if you are in the biology student, you can think of that ecology. In ecology you are use the pyramid chart. Okay, so pyramid of number, pyramid of intensity, and pyramid of like kind of things you have studied there. So pyramid is basically to depict the strength in an ascending order or descending order in the funnel chart. So pyramid chart and funnel chart are basically reciprocal of each other, where in ascending order and descending order you are going to depict the data. Okay guys, so let's move toward the very important thing, that is the heat maps and whisker. So guys, what is heat map? Heat map basically in research, this is very hot topic which can be asked in the examination. Heat maps are basically used in a single kind of, you can say, map. You are going to show different parameters or different data. Suppose there are many kind of data, and there are many kind of, you can say, the set of, suppose you are working on the rat. Okay, and then what happens, you have kind of 50 rats. How can you extrapolate that 50 rats in the line graph, or you can say in the bar chart? That will be very difficult. So heat map is a kind of thing where you can use the data from the rat, and you can show the intensity of that parameter which you have assessed. Suppose if you have assessed the like the hemoglobin level in the rat, then what will you show that there? Suppose the rat, some rat has the kind of like this, a conventional value that one has a 10, one has a 6, one has a 5. That not might be the kind of thing, because they will be similar, but that thing you can depict in the color intensity form. Suppose the darker color will show the higher number of hemoglobin, or higher number of kind of higher quantity. Okay, and the lower or lighter weight color will show the lower kind of intensity, or lower kind of number, or lower kind of the concentration. So in this way, the higher and lower concentration accordingly with the color intensity can be drawn in the heat map. And in the same way, the contour wala heat map can be used. This is the block heat map, and this is the contour heat map. Okay, contour heat map can also be used to highlight some area. Suppose if you are going to do the surface area topographical study. Basically this is the main term which you have to remember. Heat map for contour plot can be used for the topographical study. Topographical, what does this mean? Topographically basically mean the surface area से recording. Okay, so surface area वाला चीज you can record with the contour heat map. Suppose some area, if you are doing the MRI, then what happens, some area will show the higher signal. Okay, so you can do the coloring of that signal wala part, signal wala tissue, and you can highlight that area. So that area is basically will be the heat map. Okay, contour heat map basically. So contour heat map can be used to show the intensity on the topography. Okay, so guys, whisker and box plot, this is very important. Okay, so guys, please look into the figure and please remember all the different parts. So guys, what is whisker, what is median, and where is median basically? This is very important. So guys, let's look from the upper to the lower end. Maxima, minima, then in between there is a whisker. Okay, so in between these things there is a midpoint, that will be the median. So Q1 and Q3 will be the kind of quartile. Okay, so Q3 minus QR, you can see in the right hand here that interquartile range is there, IQR. Okay, this is basically the whisker and box. This is box, and the upper tail is the, and the lower tail is the whisker. Okay, so lower quartile, upper quartile, you can see here. This is very important to look into the median. Median wala thing is many times asked in the examination. So median and whisker, these two are very important. Okay, so please, whisker and median, these two things at least you can remember in the whisker and box plot. These plots are basically used to depict some of the like kind of parameters in the research, in the basically neurophysiology and in the like flow cytometry. You can use these kind of whiskers. Okay, where there is a kind of whisker. Basically, what is whisker? Whisker is basically the, you can say, the deviated part. Okay, deviated part means what happens in the data, there is the very few outliers will be there. Okay, suppose if there is the mean, that is the kind of like you can say 80, then 70 to 80 and 80 to 90 will have the mostly occupied data, or the most number of data you will find the 70 to 80 and 80 to 90 wala range mein. Okay, and the upper one, that is the near 120, you will find few data, and like 60 you will find the few data. So that is the whisker part basically, where you are going to find few data. Okay, so guys, these are the parts basically. In what happens in the quartile, this is not the case that you are going to find some outliers, because quartile is the thing where they are 25% distribution of the data. Okay, and outliers are basically removed from the data, so that the 25% and 25% wala configuration will be achieved. So guys, this was all about the, I think, lecture, and the one more important thing is the here types of study in health sciences. What happens in the AIIMS, you are going to do research, and that is basically in the medicinal field or the medical field. So guys, what happens, health sciences have different kind of studies. Okay, so guys, see, systematic review or meta-analysis is basically the review kind of study. There is not the experimental thing which is only the research. Research is basically about the analysis part also. Some analysis or systematic review you can do at your home, and that is also the part of research. Here what happens, you are going to collect all the past studies, and you are going to statistically combine the result and do the meta-analysis. So what is meta-analysis? Meta-analysis are asked many times. So meta-analysis is the analysis of different papers, different articles, or you can say different publications of the same case, or you can say same project. From the same project or from the same theme, you are going to pick up the different papers, and you are going to analyze it, and you are going to write a paper. That will be the kind of systematic review or meta-analysis. Okay, so guys, randomized control trial, where you are going to randomly assign groups of patients to receive either a treatment or a placebo. Okay, so what happens, randomized control trial. Randomized means randomly assigned group, controlled means you have a control from the patients only. Okay, and so you will have the two kind of group. One has the intervention wala group, and one has the placebo, where you have not given any kind of intervention. So you have two comparison group in the randomized control trial. Quasi experiments are basically a randomized controlled trial, but they are basically not randomized. You have just recruited the patient randomly. Okay, randomly से I mean कि you are not blinded. Okay, blinded से mean कि you know, and you know, your patients know, your guide know कि where the patients who are coming from the OPDs are basically going to be allotted. These are prior actually known. So cohort study follows a group of people to track risk factors and outcomes. Okay, so cohort means population. Basically you are having a group of population or group of people, and you are going to track the risk factor of suppose nicotine on the population, you can say. Okay, so these kind of studies where you are going to analyze the risk factors impact, that will be the cohort study. Okay, case control study compares histories of a group of people with a condition to a group of people without. So what happens in healthy control and in a patient? These two limbs will be there, healthy control and a patient, and is these two control and the healthy control and the patients will be used in the study, in the case control study. Okay, so here case and control in the study, you can like split it and remember it in this way: case and control. Randomized control means randomized patients are randomized, one to go in the intervention group and one to be placebo. Okay, cross sectional survey assesses the prevalence of an outcome in a broad population at one time point. Okay, so guys, is basically assesses the prevalence of an outcome in a broader population at a time point. So what happens here, there is a kind of follow-ups, you can say, in the cross sectional study. So case reports basically are the detailed histories of a smaller number of individual cases. Okay, so you can remember it, you can think that strength of the conclusion is basically higher in the meta-analysis and then the randomized control trial, and in the decreasing order basically at the lower part. Okay, case reports are basically least strength in the conclusive way. Okay, so guys, these three basically from upper to lower, you can say up to case control, is the observational study, cohort study, quasi and randomized control trial. These are the experimental thing where you are going to set up the experiment and you are going to do the actual research, actual experiment, you can say, in the laboratory. For review, you are not required with the laboratory, you can do at your home. Okay, so guys, this was about the types of studies in the health sciences. About sample size. What is sample size basically? Sample size is basically the number of patients or the healthy control in which you are going to do your research. That will be animal also. The animal means ki like animal में disease वाला model, or how many basically sample you are recruiting for your study is the sample size. So if a sample size is too small, बहुत कम sample size if you are going to keep, that will be, you are not able to show you the effect, or that will be not biostatistically proven or biostatistically significant. Okay, and if it will be very much large, then it will be a wastage of time and money. The main aim of the sample size calculation is to determine the number of participants required to detect a clinically relevant treatment group. So it is important to have a clinically relevant treatment and the effective thing of the kind of population. Okay, so that you can, so main ultimate goal of the research, or you can say sample size, is that you are going to show you are going to publish your journal, publish your paper. Okay, and in the paper, all the things that will be in the paper will be significantly manifested. So for significant manifestation of the data, sample size should be very, very strictly, you can say, optimum basically. Okay, so optimizing the sample size is very important, and different formulas are available depending on the study design. Please don't go into these formulas, these are very hectic formulas basically. So but remember what is sample size basically. Okay, so sample size is very important in the study. Okay, so guys, this was, I think, all about the lecture and the answer for the question, that is the father of the biostatistics is the F. Galton. Okay, so thank you so much guys, thank you so much for your kind attention. I hope you are going to enjoy the video, and please, if you want to interact with me on the social media platform, these are the list of social media platforms. Please don't forget to like, subscribe and share my channel to the needy ones. And please, all the best guys for the AIIMS PhD aspirants, and Congratulations who have done well in like kind of ICMR JRF exam and DBT JRF exam. So guys, thank you so much.